So good afternoon. Thank you all for attending this virtual lecture and meeting my friend, Dr. Valeria Aoki, who is up here from the University of São Paulo as part of a greater collaboration between our universities. University of São Paulo and Penn State University now have a formal memorandum of understanding that allows for educational exchange, which includes faculty participating at cross campuses, student exchange, and resident exchange. So if you have faculty, students, or residents interested in going to São Paulo, please let me know. Dr. Aoki completed her medical degree and residency and PhD at the University of São Paulo in Brazil. She then joined the Medical College of Wisconsin-Milwaukee for a postdoctoral fellowship in immunodermatology and was awarded a Dermatology Foundation scholarship. As I mentioned, she's an Associate Tenured Professor in University of São Paulo, also the Director of the Immunodermatology Laboratory. Her main research lines are autoimmune blistering diseases with emphasis on endemic pemphigus foliaceus in indigenous populations and inflammatory dermatoses. She's been a co-investigator of the Pemphigus Project in collaboration with the University of North Carolina at Chapel Hill since 1994. And she's been appointed as a member of the Board of Directors International Eczema Council since 2021. She served as the regional ambassador for the Academic Health Centers International Latin America and Caribbean from 2019 to 2022. And she's also currently the president of international relations at the University of São Paulo. In addition, she's a member of the editorial board of the Journal of the European Academy of Dermatology and acts as a reviewer in a number of other medical journals. Literally, she has published hundreds of articles, given hundreds of international talks, wrote dozens of book chapters, and it is a real pleasure that she has taken the time to come and speak to us about a topic for which she has a great deal of passion. Thank you, Dr. Aoki.
So thank you very much for the kind introduction, Robert. It has been a pleasure to be here for the first time, and hopefully many times will come. So I'm going to talk to you about the investigation of skin disease, which was endemic to our country, Brazil, but with unique features, and how field trips are really relevant for detecting, environmental triggers associated to autoimmunity. So this is a work that we did with collaboration with Professor Louis Diaz, Medical College of Wisconsin, and also University of North Carolina. And also to leave you a message that collaboration is essential right now in research. It has always been, but now even more, especially when we are dealing with global health. Okay, let's see. So first of all, we're talking about pemphigus. So the name pemphigus comes from the Greek pemphigs, which means blisters. So how do we classify all the autoimmune blistering conditions? We have two major groups here in North America and also in Europe. You'll see much more the pemphigus vulgaris group, which are severe diseases that initiate with mucous membrane erosions, usually oral lesions, but that can spread all over the body. However, the type of disease that we are discussing right now is the other type, pemphigus foliaceus. Does pemphigus foliaceus happen all over the places? Yes, it can be found also in different places, but it's very rare. But we do have some endemic sites. And what's interesting about those endemic sites and how does an autoimmune disease can become endemic, especially in a tropical country? Do some environmental factors, including vectors of infectious disease, interfere with the course or even the trigger of the disease? This is what our research will try to explain to you. So pemphigus foliaceus then happens all over. The classic form, it's called Casenavis pemphigus. If you read the text, the classic dermatological textbooks, you see this classic form, Casenavis, which was detected by the end of the 19th century. So the type of pemphigus we're going to discuss here, it's called endemic pemphigus foliaceus. And in the literature, it has also a Brazilian nickname, but it's now, you know, all well known, even in the scientific world, which is Fogo Selvagem, which means wildfire. Why? This was the symptom that all the patients complained when they got the disease. Your skin will burn as fire. So we have some other variants of pemphigus, of course, but we are going to focus on endemic pemphigus foliaceus. So where can we find this disease? It's rare, but the main focus is Brazil, where I come from. So it was first reported in 1903. And look how difficult it is to detect this disease, because it can, you know, resemble other diseases. When the first cases were reported, they were not reported as autoimmune blistering conditions. They were reported as superficial mycosis, because the clinical features are similar. And I'm going to show you some pictures about those cases. You may find some reports also in other parts of South America, especially Peru, Colombia, Ecuador, Venezuela, and Paraguay. But there's another outbreak in Tunisia that happened in 1995, where the first reports also showed endemic places where you could find the disease, but with a little different aspects, especially in the epidemiology. So you see, it's a very restricted disease in terms of geographic distribution. So what's different between the classic form and the endemic form that we notice in our country? First, if you clinically evaluate a patient, there's no difference. You cannot tell who is an endemic form or who is a classic form. So what's the difference? The difference is, first, you have geographic sites involved specifically for the endemic forms. And second, it's an autoimmune disease, but you have many members of the family, so blood-related members with the disease. And third, it will affect much more children and young adults. If you consider the other autoimmune conditions, the blistering skin diseases, they won't, usually they show up above the age of 50. So if you think of epidemiology, you wouldn't think of pemphigus foliaceus in a young patient if you're here in North America. That's the first point. Okay, so, and between the two sites, the Brazilian site and the North African site, do we have differences? A little bit. In Tunisia, you have young female adults as more prevalent cases. In Brazil, you don't have gender differences, but they're both young patients. And because you have familial cases, in the past, when we didn't know about the etiology of this disease, people would think that it was contagious. So many patients in the beginning, let us say 19th century, beginning of the 20th century, they would consider it an infectious disease, and patients would be released by their own because they didn't have the treatment, they didn't have the diagnosis. So it was a very dramatic condition. So if a patient comes to your office, how can you suspect of pemphigus foliaceus, the endemic form, or even the classic form? So this is a lady. What can you see here? You see this very symmetric bilateral erythematous and desquamative plaques, right? Bilateral on her face. And here in North America, what would you think of this lady? You think of lupus, right? It should be your first hypothesis. Why? Because it's a sun-exposed area and you have those kind of lesions. But you don't see blisters here. So how can you think of pemphigus foliaceus? Later on, we will have to add additional laboratory diagnosis to confirm that this is a pemphigus foliaceus. So patients usually show up in your office like that, with localized lesions, where in sun-exposed areas, mostly on the face, and also the anterior facial trunk. What happens to this patient? It can they can evolve into a spontaneous remission, as any other autoimmune or inflammatory condition, or it can become generalized disease and severe disease. In the past, about 90% of those patients would die because of pemphigus foliaceus, because we didn't have anti-inflammatory drugs or potent immunosuppressant drugs. Right now, fortunately, we have not only the regular immunosuppressant drugs, but also the immunobiologics. We use rituximab, for instance, an anti-CD20 to control the disease. So mortality rates really are 5 to 10%, and usually they happen because of, secondary infection. Also, when a localized form heals, it can also become a chronic form. It's not that severe, but it's really interferes with the quality of life of the patients, right? And they come from rural areas, and sun exposure will enhance the disease. So in terms of quality of life and their profession, usually the disease interferes a lot. So it's very complicated. They sometimes have to change their profession because of the sun exposure, that it's not recommended. Another form that can show up for the physician, and it's tricky. If you're a dermatologist, it's a little bit easier, but like for GPs that don't see those cases, sometimes the patients show up as erythroderma, which is an erythema and scaling all over the body. So these are the generalized forms. They may start localized, but this is on your left side, bullous exfoliative severe form in a kid. This kid, she was a 9-year-old girl, and this one was a teenager girl that came to our office, and she had this erythroderma. So you can see squalene and erythema all over her body, very severe cases in young patients. So here, this is the diversity of pemphigus foliaceus, clinically speaking. What would you think of this patient on your left side? You see those circinated lesions here, kind of tinea imbricata-like. So this was very similar to the first cases reported in Brazil. They looked like tinea, superficial tinea, but in fact, these were all blisters or vesicles caused by the autoantibody deposition on the skin. Other forms are also common. You see here pemphigus herpetiformis. In this case, the patient has the blisters plus a very prominent inflammatory response. And here, do you see here erythema and edema? This resembles another blistering disease, which is bullous pemphigoid. This is a differential diagnosis. And bullous pemphigoid, this disease you see here a lot in North America. So these are some difficult diagnoses when you face this condition. And the third patient here shows a hyperkeratotic form. In this form, what do you see? Do you see any blisters? Not at all. You see hyperkeratotic, hyperchromic lesions. This look like a seborrheic keratosis, right? But this is a pemphigus foliaceus. So these are variants that are hard to detect. And what's intriguing about this disease? So I'm going to talk about the genetic factors, the immunological factors, and the environmental triggers that are involved in this skin disease. So our story here starts back in the beginning of the 90s. So at that time, I was doing my postdoc fellowship with Dr. Diaz in Wisconsin, and a physician from Brazil, a dermatologist from the local university, called us and said, I think I have something interesting for you guys to study. I have a focus of endemic pemphigus foliaceus, and it's very hard to detect a very, a specific site. We used to track patients all over the country, but not in a single site. And it happened to be an indigenous reservation, where in Brazil, in the state of Mato Grosso do Sul, which is the beginning of the Pantanal region, almost in the border of Bolivia and Paraguay. These are Tirena Indians. This was the photographic here documentation from area view. So it was at that time a very segregated population. We had to get all the permits possible for studying indigenous population in Brazil. And look what we found. We had to start from the very beginning. Okay, we have a focus of an endemic disease, but the patients, they didn't have the indigenous population, they didn't have register, they didn't have, all the mapping where they lived. So how could we start an epidemiologic study here without all this data? So we started collecting all the pedigrees and mapping all the housing. And look what we saw. This is a house from the 1942 time where the disease was, clothing the state of São Paulo. Now, São Paulo does not have this disease as an endemic form because it's urbanized. So here in that region, 50 years later, we found the same conditions than in 1942. So a perfect habitat for studying the natural course of the disease and all the etiology triggers. This was the first point. Second point, did we find familial cases? This is a unique feature of this disease as being classified as endemic. Yes, we did find, at that time, we found 3 generations of patients with the disease. Right now, we have 5 generations, almost 30 years later. So we mapped everything, and we had a very high qualified geneticist with us. And what we detected was that patients that had this sequence of 8 alleles in the HLA-DRB1, with those sequences, had a relative risk of 14 to have the disease. Was this the only condition? No, but if you have the sequence, you are really prone to have the disease if you lived there since you were a kid. So the immunological factors, how can I prove that this disease is autoimmune? So the classic form of Pemphigus foliaceus, we know that the autoantibodies were detected in the 60s by Dr. Jordan and collaborators. And then the endemic form was detected years later. So we know that here, this is the skin condition. So you see superficial blisters. So when the patient arrives to you, you won't see blisters anymore. Why? Because the blister is very superficial. The cleavage here is called subcorneal. It's really 1 or 2 layers on the roof. That's why you see erosions or crusts, right? And there's a hallmark of pemphigus, which is acantholysis, which is the detachment of the keratinocytes. They lose their form because they lose their connections, and they become round cells here, and this is called acantholysis. It happens in all forms of pemphigus. Oops. It's not pathognomonic, but it's very suggestive. And here, in those patients with the endemic forms of Pemphigus foliaceus, they also showed what? Here, the deposition of autoantibodies on the skin and also circulating autoantibodies. And the autoantigen is desmoglein 1, which is a protein that's located in the core of the desmosome. And it weighs 160 kilodaltons. So this was about in the 80s, mid 80s, that we detected all these features of the disease. But I think that the beauty of the study happened in this village. Why? Because I think I told Rob the story, but I was there doing my PhD, and Dr. Diaz said, well, we are testing a new antigen that we're going to perform later on, ELISA plaques, to have a very feasible diagnostic tool for those patients. I said, fine. So we prepared the antigen, which was desmoglein 1, recombinant proteins. At that time, we were initiating this technique. And look, all the patients with Fogo Selvagem, the endemic forms, reacted beautifully against the protein. So I was really happy. But what happened on the next day, I used controls. And I thought, well, the best controls would be the people living in the endemic areas that are not sick. They don't have the skin disease. But then turn out that 50% of the people that lived in the region reacted against my protein. So I had here two options. Either my antigen was bad, so I would have to produce other antigens, or something was happening in their autoimmune response with the normal population. Why were they reacting against desmoglein 1? And why didn't they have the disease, even though they were under the same conditions? And maybe they would have the same genetic alleles. This study was published in the New England Journal almost, oh my God, it was year 2000, a long time ago, but it was beautiful because we proved that if you are more distant from the endemic sites, the normal population wouldn't react against desmoglein 1. So the problem was not the antigen. There was something happening in the region that the local population without the disease was producing autoantibodies. So we had to detect then the environmental triggers that could collaborate to this response, right? Autoimmune, but was not provoking the disease. So the next steps would be then doing epitope mapping. And I think we got part of the response. Why? Because we kind of sophisticated the epitope mapping, and then we saw that normal population living in endemic areas would produce autoantibodies belonging to the IgG subclass. And they would recognize only the extracellular domain of the protein, which is not pathogenic. But if you have the correct allele sequence in your HLA-DRB1, and if you are exposed to constant environmental stimuli, you can shift the response, provoke what we call intramolecular epitope spreading. So you start seeing other parts of the molecule, and you shift the IgG1 to the IgG isotype response, and this will provoke the disease. So this was the model, the concept of how a normal population and a sick population will differ in terms of autoimmune response. This was a beautiful study published by our group, and Dr. Ning Li, who's now a professor at UNC, she was doing her PhD as well. So this was really a good discovery. And then we had to proceed to investigate a little bit of the autoantibody response if we had the hypothesis that environmental triggers would be involved. So when you think about insect bites, we think about IgE response, right? Or sometimes IgM and also IgG4. So what happens? Do I have also antibodies that belong to those classes that are not only IgG that react against desmoglein 1 in that population? Yes, so we proved that Pemphigus foliaceus patients in endemic areas in Brazil do have IgE, IgM, and also IgG4 antibodies against desmoglein 1. So maybe, and then later on, we used some epidemiological models, and we detected that IgG4 was a good predictor of future disease. And those normal people who had IgM could be, a population with a serological marker for the endemic forms of Pemphigus foliaceus. Because at that time, we didn't have anything to differentiate, rather than epidemiology, the classic forms of Pemphigus foliaceus versus the endemic forms. So this was another interesting finding. This was published in 2020. We had a collaboration with Columbia University. So Dr. Diaz wanted to know the exact location of the binding of the antibody with the autoantigen. Why is this important? Because now we are in times of precision medicine, precision therapy, and novel molecules have been detected, right, to be used in those autoimmunity conditions or inflammatory conditions. So we have to know exactly where the autoantibody binds the epitope. So according to the study, if we make shifts in the extracellular domain 1 of desmoglein 1, we're able to, inhibit the autoantibody response. And these were the sites where the epitope change could influence, and maybe if we produce some molecules that could have this different configuration, we could block the autoimmune disease. And these are the alleles that we have to interfere if we want to block the binding of the antibody versus the autoantigen. So this is the very interesting part. Our family doctors do some field trips. When we started to investigate environmental triggers, the old researchers always had the hypothesis that a vector could interfere with the disease. How? Maybe, through constant insect bite stimuli, you could probably do antigen mimicry with desmoglein 1. You could modify the skin protein. The insect could inject some anticoagulant substances that would change our protein. And what we did, first, we did an epidemiology study proving that the types of housing in the village would interfere with the risk of the disease. And second, we thought, well, Brazil is a country where you have a lot of infectious disease, such as Chagas disease, leishmaniasis, and these are diseases that have hematophagous insects as vectors, right? And we detected that people who have those infectious diseases reacted against desmoglein 1. So if you see here, where do we have Chagas disease? There's a kind of similarity in terms of geographic distribution of infectious diseases with hematophagous vectors and Fogo Selvagem. So we had something here, right? But unfortunately, when we were testing against Trypanosoma cruzi, the Chagas disease factor, we didn't find anything. And we extracted thousands and tons of salivary glands from those triatomas, and with no similarity with desmoglein 1. But finally, about 15 years later, we moved then to phlebotomus, which is the vector of leishmaniasis. And see, the distribution of leishmaniasis can be also somehow similar to endemic pemphigus foliaceus. And finally, we got a response. So if you extract salivary glands and then you detect LGM11 and LGM17, these are two proteins that you may find in the salivary gland of the phlebotomist. They correlate with desmoglein 1, and patients with the disease recognize both desmoglein 1 and also the salivary gland proteins. So there's a sequence homology between those salivary glands and the human desmoglein 1 ectodomain. So here we are proving that somehow our hypothesis of antigen mimicry can be true, right? So in summary here, so you have endemic areas with a lot of exposure of insect bites. Second, the salivary glands from phlebotomists, such as LGM17 and LGM11, they have antigen mimicry with desmoglein 1, an autoantigen. We have desmoglein 1, right? So this may provoke an autoimmune response. Third, you have to switch from a regular IgG1 response as a normal person without disease to IgG4 if you have the correct sequence of HLA-DRB1. And with this, we can probably attribute LGM17 as the major endemic Pemphigus foliaceus autoantigen. And this is just to conclude, it's interesting because if you think of phlebotomies, immediately you think about leishmaniasis, right? So you have here a vector that is common to an infectious disease and to an autoimmune disease. So here's the beauty. A phlebotomist can bite a human being and it can inject, an infectious vector and then infectious parasite, and then you will have leishmaniasis. But an insect like that can bite a human being and then provoke an autoimmune disease. So you have the two faces of the same factor. And why is this important? Because of that, those salivary gland proteins are used for producing vaccines against leishmaniasis. But you cannot use vaccines produced with those proteins in those countries where you have endemic forms of those diseases. So this is important if you consider global health and regional distribution of some conditions. Okay, so I'll just finalize my talk. This is almost 30 years of collaboration with the University of São Paulo, the University of North Carolina, Chapel Hill, and the Federal University of Mato Grosso do Sul. And to thank all the indigenous tribes, populations that really collaborated with our study, and hopefully it will serve as a good model for autoimmunity and environmental triggers. And this is a thank you note. So Dr. Wen and Dr. Lennon were here in my country, Brazil, and I do hope that more collaborations will arise after this initial projects. Thank you very much.
Thank you so much, Dr. Aoki. Does anyone have questions or comments about the process, the science of the talk?
I would love, yes, dermatologists. Yeah.
Well, researchers in the room, not dermatologists, but I'm curious if, horses and dogs get Pemphigus foliaceus also. Are there any examples of that happening locally, endemically, or is it very specific, the spread to humans?
We try to investigate some of the animals, not only dogs and cats, but also cows, because in the cows there's a lot of desmoglein 1, but we didn't find any animals with the disease. So and all the work with the vet colleagues, they were done in animals coming from non-endemic areas. Usually they were from the capital, and we didn't find so far differences. We ran IgG subclasses in those animals, and I don't think they have different patterns different from humans, but like other mammals wouldn't be, let us say, reservoirs or different models for this autoimmune model. Any more questions?
Yes, thank you, Dr. Aoki, for a great talk. It's very exciting from the perspective of both science Dermatology and Global Health combined. I'm with Drs. Lennon and Tuan in Family and Community Medicine. Thank you so much for coming. It's an honor to have you here on campus. From the perspective of family medicine and global health combined, I have a question about what do you think are the next steps in this research, both from a kind of a practical point of view and any kind of applications in global health of this research?
Well, I think the next steps would be mapping more regions that have this endemic forms. Like there are other countries in South America, as I mentioned, so Peru, Colombia, Venezuela. We don't know about their endemic triggers, if they are the same. They also do have a lot of leishmaniasis there, but we don't know this response. So next steps would be expanding the same model to other countries where you have this endemic forms, and also trying to see whether the non-endemic forms would respond well. Do you remember the blocking of and modifying of the autoantigen? If this would work with the classic forms of the disease,
right?
Because we're thinking here in probably targeting new molecules, new sites for new molecules to have a specific precision treatment. So this would be the next steps.
Do you have a sense of the range of this in work that I have done in villages in Honduras? We saw actually a lot of leishmaniasis, not pemphigus as much, but for sure evidence acute and chronic actually of leishmaniasis. Do you think do you have a sense of the full range?
I have no sense of the full range, but there are some hypotheses, David, that perhaps because the other question would be, well, you're saying about the vector of leishmaniasis, but what happens? Don't you have patients with both diseases?
Yes, of
right? And it's rare. It's really rare to see the association of leishmaniasis with pemphigus. So maybe you may have a protection. If you have one, you won't have the other. You may have the same vector triggering some things, but as I said, you can go to different directions. But we have to expand this. This is just, one site that could serve as a model, but we still have a lot of work to do.
Interesting. Thank you.
Robert, any comments?
Yeah, well, to me, I think that's a particularly clever way of looking at this. Is you have a model explanation in your area. It would be interesting to find other areas with high prevalence of leishmaniasis. So some pockets in the Middle East where there's a lot of it. It might be very interesting to look at their genetics and see, okay, what do they have or what are they missing such that the disease doesn't progress? And I suspect, just as you've intimated, there might be more than one trigger. So the desmoglein antibody formation is probably one step, and there might be another one still to ferret out. But it's fascinating to see this in real time. Fascinating to see this process over 30 years to identify this. It also gives us, from a global health perspective, a phenomenal appreciation for the concept of One Health, where we don't look at human health in a vacuum, but we look at the patient and then we say, okay, what else is touching the patient? Insects, local flora, other local fauna, the water supply, the air supply, all of these things integrate into the ultimate health experience of the human. And in human health, that's poorly defined. So it's wonderful to see this as the beginning of a map where you did the map of all of the people. So now you have this wonderful genetic background, and now you can look at, okay, well, now let's look at the insect impacts, and then we can add food, water, everything else. And it really gives us this picture of why One Health is such a valuable construct, leading back to David's concept of what are the next steps. Well, if we can figure out that constellation, we can fix the one part by saying, all right, in other parts that have, 3 of the 4 things that are required, there's no disease. So instead of focusing on blocking all 4 things that are required, we can just take the one thing away, right? For example, in this case, you can get rid of most of the insect bite induced autoimmune response by getting rid of the insect bites. Right, that's what urbanization, the benefit of urbanization was, you got rid of that. Okay, so maybe we can go to these tribes and say, well, we can't fix the underlying problem, but we can give you screens. We can give you some DEET. We can give you some screens. We can talk about avoiding the insect bite. So I think it's a fantastic example of the power of One Health.
And it's more interesting because in that region, in the 70s, there was a lot of malaria. It was a malaria region, and the federal government really control the insect spread, with all kinds of DDT and those substances in the past. So all the insects were removed. And later on, probably the phlebotomists arrived because we didn't have leishmania at that time in that region. We had malaria, but then there was a switch of the type of insect. Yeah. So we cannot think only about, the patient itself, but as you said, all the components of this environment.
Are there any other questions or comments?
I just wanted to say to the young researchers, never give up. Because in the beginning, we started with black flies, and I told just a sum up of the story, but the first insect that we went through the salivary glands were black flies, because there was a lot of black flies in the region. And one of our entomologists detected that in the Brazilian coast, where you don't have the disease, we also have a lot of black flies. And it turned out that the species of black flies in the coast was different from the ones in the village. And then we said, oh, that's the answer. And we were dissecting salivary glands from black flies like crazy for 1 year, but all the experience went wrong. But we pursued, and one day we found out something that related to desmoglein 1.
Fantastic. And if anyone listening is interested in collaborative relationships with researchers at University of São Paulo in certainly dermatology, but any other field, by all means, let us know. The collaboration between our institutions was formalized last fall, already growing by leaps and bounds. We've had a handful of our medical students go down. We're looking for resident rotations down there as well, and we're hosting a handful of students this year. Dr. Aoki is the second researcher to be invited to our institution as part of this collaboration. And Dr. Tuan, who's on, has been invited down to Sao Paulo as a visiting scholar to do collaborative work. And I've been finishing up my Fulbright Scholar Award there as well. And we're hoping Dr. Rabago will join us for the remote health check experience at the end of June. And we'd like to invite you. You're welcome to join. It's an incredible experience. It also has academic implications, because doing collaborations like this, by definition, gives you the international recognition that you need to move up the professorial ranks. And with that, if there's no other questions, we'll let you go. Andrew, we'll be over there directly.
Thank you.